This initial lab scene is the virtual lab for doing the simulation work to inoculate, incubate, and interpret the results of biochemical tests used to study bacteria. However, there's more to the virtual lab, and these other features are accessed by the links found above the virtual lab. You will note that as you begin, the lab link is highlighted, indicating you are in the virtual lab. Let's look at the other links. Select Session Box. This drop-down box lists all unknown microbes you have created or that have been created for you by your instructor. Click on the drop-down box and select one of the unknowns. Its name will appear in the box as the unknown you are currently working on. If you need to switch to work on a different one, you simply select the desired microbe from the list. ID Matrix. There are 124 microbes and over 50 tests included in the virtual lab. Figuring out which tests to do for identifying a microbe is accomplished by comparing the results from a biochemical test with the known results for all 124 microbes. When a positive result for test A is recorded, all microbes with a negative result for test A are eliminated from the matrix. As you do tests, the number of microbes still possible is reduced, until only a single microbe remains, your unknown. You can click on this link to see where your ID process stands and determine which test to do next. Right now, the link is not active because no unknown microbe has been loaded into the system. Everything you do in the virtual lab is noted by the simulation. Every test done, every result recorded, every mistake made, the amount of time spent working, all of this is recorded in the lab report. You will be able to see some of this as you work, but other notes and errors will not appear until you have provided an ID for the microbe being worked on. This is valuable feedback for you and your instructor as you hone your micro lab skills. Unknown. This link has many options of importance to starting and ending the bacterial ID process. Click on the New option and you will be taken to a form for creating a new unknown microbe to work on. There is no limit to how many microbes you create. These will appear in the Select Session box at left. Once you find the identity of a microbe, you click on the Identify option to enter it into the system. You will be told whether you were right or not, and the lab report will now report all the details of your work. Under these two options are resources allowing you to see the case study for this unknown. It was shown to you when the unknown was first created. And also the gram stain. Your gram stain result is your first test result and will reduce the number of bacteria listed in the ID matrix significantly. Tests. This link provides access to the resources needed to do biochemical tests for studying a microbe. Select Media takes you to a list of all the media appropriate to the study of the bacterium you are working on. Add Chemicals brings up a list of chemicals, stains, and reagents used in the virtual lab. Some tests require addition of a chemical called a reagent after the culture has grown overnight to determine the presence of a growth product. Likewise, some of the chemicals listed here are stains used in the gram stain process. Find those chemicals here. Videos. Some of the tests important to ID of bacteria are best demonstrated by use of a short video. The video-based tests relevant to the microbe being studied are listed here. Clicking on the link takes you to the video. The activities and help links will be described in separate videos.